Okay, so I'm gonna go ahead and give you guys a little genetic practice test so you can better prepare for my test and better prepare for your day school test. So okay, so first all credit goes to Rosedale Academy. I did not make this. So first, the product meiosis 2 is haploids, pretty straightforward. Alleles for the same trace separate drawing meiosis 1, pretty straightforward. I refer to the illustration above. The cell in diagram 1 is anaphase, breaking it apart. Mitosis ends with the stage with not this. This is at the start. Not this. That's metaphase. Anaphase. Telophase. I'll say that's cytokinese. Or is that the spindle fibers forming? Okay, we'll give it a 2. So I would say okay, it's not 2. Then it will be 4. Sure. I'll give it. Oh, mitosis end. Okay, so it's 5. My bad, because that was telophase. So 5. Which is B. How many possible phenotypes? Okay, phenotypes. So it could only be type A completely or AO. So 2. Well, phenotypes, I guess you could say 1 because AO is a genotype, but I guess. So test cross is performed by crossing a dominant individual with an unknown genotype with uh dominant individual. Okay, so you usually cross it with a homozygous recessive individual. This way you could right away tell the unknown genotype because if your kid if the result has around 50% recessive kids and 50% dominant kids, then we know this person is a heterozygote. If all of the kids are fully recessive, then that means the unknown genotype is homozygous recessive, and then everything is dominant, then this unknown genotype is homozygous dominant. Pretty straightforward. In this cell cycle, the cell prepares for mitosis by duplicating the cell's DNA. This is S phase, the synthesis phase where the cell grows. Which of them is not an advantage of asexual reproduction? It's fast, so that's an advantage. Doesn't require special reproductive structures. That's also an advantage. Increases diversity in a population. It actually doesn't. So we're going to leave it there. Conserves energy. That's an advantage. So the only one we have is C. In a cross between two heterozygote purple pea plants, 1/4 of the offspring will be white. This is because the plants will still carry the gene for the recessive trait, it just doesn't express. Okay, that's correct. Genes are stored independently on the metaphase plate. That is not true. The alleles segregate during meiosis, and each parent can give their either allele in a gamete. Okay, so A and C are both correct. Harmful X-linked trait determined by a recessive gene tend to appear only in females, which is not true because X-linked is more common in males. Do not skip generations, which is also not true because recessive genes always skip generations. Are usually not passed from father to son, which is true. Are usually passed by only carrier males, which is wrong. So they're never passed from father. An X-linked trait, their dad can actually never pass it to their son, simply because the dad will always pass the Y-linked trait. There's no way you're gonna have a son with 2 X chromosomes. That would be a girl technically. I wanted to skip the labeling, and you can skip the labeling. I'm not gonna put too much labeling on my test as well because really it's not that good of an imagery, and it makes people confused. Especially, hey, what is the difference between this and that? Like you can't tell it. Okay, so do you expect the rate of cell division to be higher in an adult or child? So I'll give you a second to think. Okay, so the correct answer is child. So, and your reasoning should be children undergo rapid growth and development. This is because they just came out of the womb, the uterus, and they often require much, much, much more frequent cell division so they can actually grow some tissues, grow organs, grow bones, teeth, skin, whatever. So adults primarily only use cell division for maintenance and repair. So there's nothing too big. There's no big construction for them to do besides just repairing. So it's a lot higher in children's. Errors that occur during meiosis are present in all cells of the body, whereas errors that occur during mitosis may occur in only a small number of cells. Explain why this happens. Okay, so we're gonna talk about the nature of mitosis and meiosis, I guess. So meiosis produces gametes, which is potentially sex cells that pass it on and go on to generations. Uh, these errors, also known as non-disjunctions, they will affect all the offsprings, and then the offspring will affect the rest of the offsprings, and it just kind of stacks on to dominoes effect. Mitosis are in somatic cells. So mitosis, the errors in mitosis, they only stay in a limited amount of cells. They only stay in the daughter cells, and some of these daughter cells will die, and it's a much like it's a less spread because you realize for somatic cells, it's gonna only stay in you. Like if I had an error in the mitosis, I'm gonna pass it on to the kids. But since I'm doing mitosis, the kids will not pass it on anymore. That's it. It's limited. It's also called It's also called localized mutation. Okay, so let's talk about this. So the somatic cells in a horse have 64 chromosomes. What is the diploid number for a horse? What is the haploid number for a horse? How many chromosomes are present in a normal gamete? How many chromosomes are present at a cell at prophase 1? Okay, so the diploid number is 64 because chromosomes, right? Haploid number is 32. How many chromosomes are present in a normal gamete? Okay, so the normal gamete should have 32, so because it has half, and prophase 1 should have 64 since the chromosomes got duplicated, but they're not separated. Okay, pretty straightforward. Two parents have the same phenotype for a given trait. Produce a child with a different phenotype for the same trait. Use a Punnett square to explain this observation. So I'm not gonna write it out for you. And no, the parents did not cheat. I hope. I I hope. Yeah. So the thing is, these parents are likely heterozygotes. Well, in fact, definitely heterozygotes, or the mom cheat. But so the parents are heterozygote. They're both big A, small a. So big A, small a is gonna give out 25% to be big A, homozygote big A, and a 50% chance to be heterozygote, and a 25% chance to be recessive. So if the kid has a different phenotype, so the kid is likely completely recessive. So 25% chance, it's okay. Let's go on 16. The allele for black hair, big B, is dominant to the allele for white hair, small b, in guinea pigs. Assume two heterozygotes are crossed. So write the genotypes of the parents, which is big B, small b, big B, small b. Punnett square. So it's the same Punnett square that we used last time. Very basic. 25% to be 25% to be black. Oh no. 75% to be black and 25% to be white. So the probability. Okay, so 25% to be white. Just said that. And a farmer crossed a black rooster with a white hen. Of the 7 offspring, 3 are black, 3 are speckled black and white, and 1 is white. What can you infer about the inheritance pattern of the alleles on black feathers? So we are talking about I guess the special one in this case. So this is a codominant because they're both expressed at the same time. So the parents, which is the black rooster and the white hen, it's likely a BB and WW because we have 3 black and 3 white, and we also have 3 speckled white and black. So incomplete dominance. This is an incomplete dominance. Okay, no, this is not incomplete dominance. This is codominance. So big BBWW, it's gonna produce BWBW, right? So BWBW, the kids are going to be yeah. So BWBWBW, yeah, okay, speckled, and then could be white, yeah. So the inheritance patterns of this is codominance, and it's a simple. Oh no, sorry. It's incomplete dominance, so BB and WW. So the parents should be BW and BW, which is okay. No, this question there should be something wrong because you can't have it. Yeah, you can't have it. This is wrong. So the inheritance path. This question is wrong. Skip it. What that? Given the inheritance, what are they expected? Okay, so also gonna skip that, I guess, because it's wrong. I just realized it's not possible to have to if your parent is fully black and your parent is fully white, then you will always pass on the B trait, and the rooster will always pass on the white trait. So that doesn't work. So let's go on to 18. A variety of reproductive strategies, technologies, including cloning, artificial insemination, and in vitro fertilization, are used to control genetic diversity of farm animals or plant crops. Choose one method. So I'll choose artificial insemination. So this is semen from genetically superior male is collected and inserted into a female. So this controls diversity as we are using desirable alleles. For example, disease resistance, high yield across populations. This reduces genetic variability by promoting specific traits. This avoids random mating and concentrates selected genes. Not that good in this case. Genetic testing has many significant bad benefits. However, ethical dilemmas are also associated with it. Write a paragraph explaining why you're either for or against prenatal genetic testing. If you found it difficult to support only one side of this issue, okay, explain why. So I'm gonna say it's good as long as it's ethical with some ethical standards. So this actually enables early intervention for conditions. So whether you have hepatitis B, A, whatever, and you can actually detect that, and then the family they will be like, okay, I know what I'm going up to. Do I still want to have the kid? Sure. If I don't want to have the kid, what am I gonna do? So that is first, and then uh, if their kids caught like a very rare disease, and then it takes a lot of money, it could financially prepare them, reduce some uncertainty. However, if there is absolutely no ethics in this is probably misusing because there's a lot of psychological stress from the results or pregnancy termination based on like if I test it and I realize, hey, look, the kids is not gonna be so smart, and the parents is gonna kill it. That's not good because it's still a person. Even though it hasn't walked out of the uterus yet, it's still a person. So this is something I really want to talk about is that are they if they haven't been if they're not born yet, are they really not a person? Like no, they're still a person. So I feel like there's a lot of things that's way more to this, and I think overall it's good, especially at main hospitals. Strict regulations must be needed to prevent any discrimination and ensure that this thing is actually working. However, I do think this is a beneficial thing because I'm always up for technology and improvements. And with that said, that is our last question. So I mostly want to know. So I'm gonna put. So for my test, I'm gonna put a lot of short answers. So expect like maybe 20 short answers and 10 multiple choice, and maybe one diagram question. And that's pretty much it.